The question we'll address in this session is gene expression, going from information in DNA to synthesis of protein. This occurs in two stages. The first stage is called transcription, which is the process of synthesizing RNA based on information on DNA. So the information in DNA is transcribed or copied into an RNA molecule. And we'll talk about how that happens in bacteria. And in eukaryotic cells, the additional complications involved in RNA processing in the nucleus before the RNA transcript can be translated to protein. This process is called the central dogma of molecular biology, that information encoded in DNA is replicated by DNA polymerase, and we talked about that in the last video. The information in DNA then can also be transcribed or copied by the enzyme RNA polymerase into RNA. We call this transcription because the information in the RNA is a sequence of bases that is identical to the information in the DNA, which is also a sequence of bases. The only difference, of course, is that wherever the DNA contains a T, the RNA has a U. Then from RNA, which is a sequence of bases, that information is now translated into a sequence of amino acids in a polypeptide chain. Because now we're going from a sequence of nucleotide bases to a sequence of amino acids, it is like you're going from one language to a completely different language, and this is why we call it translation. Finally, and we don't have time in this video certainly to cover this process, the interaction of proteins with the environment, the interaction of proteins also with some non-coding RNA eventually leads to the phenotype of the organism. We'll start with transcription. The enzyme RNA polymerase binds to the DNA, and it binds to the DNA not just at random places, but at specific places called promoters. So the definition of a promoter is the site in the DNA where RNA polymerase binds to start transcription. What's shown here is a promoter used in E. coli. If this is the site where transcription starts, if you see upstream of this transcription start site, there are a sequence of bases, around 10 bases upstream and 35 bases upstream, that are characteristic of E. coli promoters, and E. coli RNA polymerase, with the assistance of a transcription factor called a sigma factor, recognizes these sites in the E. coli chromosomal DNA as being sites where it should start transcription and make RNA. As RNA polymerase binds to the promoter, the base pairing between the bases in the promoter becomes undone. The DNA molecule unwinds a little, and this creates space for RNA polymerase to bind to the template strand. Note the labeling of the two strands of DNA. The lowest strand here is called a template strand, and again, by definition, the template strand is the strand that RNA polymerase reads, and therefore the RNA molecule shown in blue, or the transcript, is complementary to the template strand. The RNA molecule actually has the same sequence of bases, you will notice, as the coding strand of DNA. Both the coding strand of DNA and the RNA transcript, or the RNA molecule that is transcribed from the template strand, they are both complementary to the template strand. I also want to point out that RNA polymerase synthesizes RNA from a 5' to 3' direction, reads the template DNA strand from a 3' to 5' direction. In that sense, it is similar to the activity of DNA polymerase. The difference between DNA polymerase and RNA polymerase is that DNA polymerases require a primer. RNA polymerases do not require any primer to initiate transcription. Once the RNA molecule is finished being made, it can be translated. The process of translation requires a number of different players. The most important, well, okay, they're all essential, so one isn't any more important than the other. So one of them is the ribosome. What's shown here are the two subunits of the ribosome, the large and small subunits, docked together. Now in the cell, these two subunits are docked together only in the presence of messenger RNA and only in the process of translation. If there is no messenger RNA, these two subunits are separated. These are large complexes of both RNA and protein. So each subunit has a ribosomal RNA plus lots of proteins. In all, the two subunits together have 3 ribosomal RNA molecules, and in E. coli, over 50 different ribosomal proteins. They all assemble into this rather large complex, and the ribosomal RNA molecules comprise an important portion, certainly by weight as well as by volume, of the ribosome, and the ribosomal RNA plays a key role in forming the catalytic site of the ribosome. Another key player is the transfer RNA, also known as tRNA. So tRNAs are also called transfer RNAs. Because they bring amino acids to the ribosome, and they bring the right or deliver amino acids to the growing polypeptide chain as specified by the messenger RNA that the ribosome is translating. So the tRNA molecule is typically drawn in this clover leaf shape shown here in the left. It's a single RNA molecule that has internal base pairing. So the bases within the RNA form hydrogen bonds with other bases in the same tRNA molecule, and these base paired regions are called stems. And in addition to the acceptor stem, because this acceptor is, it's called the acceptor stem because this is where the amino acid is covalently attached right there to that hydroxyl. And then in addition to the acceptor stem, there are 3 other stems, each with a loop. This loop is a sequence of bases that's not paired. Okay. The middle of these loops is called the anticodon loop, and these 3 bases form the anticodon. The anticodon is complementary to the codon on the messenger RNA. This is what the folded tRNA looks like in a more 3-dimensional view. And again, here is where the acceptor stem is and where the amino acid is attached. If you think about it, the cell has to have a system for making sure that each tRNA gets the right amino acid attached to it. And that's the job of these family of enzymes called aminoacyl tRNA synthetases. Each one binds a specific tRNA, so the tRNA here is shown in this ball structure, and the enzyme is shown with just the peptide backbone. So these tRNA synthesase enzymes, each of them recognizes the correct tRNA by its anticodon loop. So there's a part of the enzyme that interacts with the anticodon loop of the tRNA. And then another part of the enzyme binds the correct amino acid for this tRNA. In addition, it uses energy from ATP hydrolysis. So here you can see where the ATP is bound. And this enzyme will hydrolyze ATP to AMP plus 2 phosphates. And that energy is used to covalently attach the amino acid to this 2' hydroxyl group of the tRNA. This tRNA charging system, so we call a tRNA charged when it contains the amino acid attached to it. This tRNA charging system is the basis of this universal genetic code. So this is a codon table. You don't have to memorize it. We will supply this exact same table on your exam. I just want to point out a few things. First, there are 64 possible codons, and that's because each codon consists of 3 bases. There are 4 bases, 4 different possibilities for each of the 3 positions in a codon. So 4 times 4 times 4 gives us 64. And you can read this table by identifying the first base here, the second base here in these columns, and then within each of these boxes, the third base. Each of these boxes then gives you 4 lines based on the third base. Okay? So I also want to say or point out that this code is what we call degenerate. Okay? And what that means is that each amino acid, there are only 20 amino acids and 64 codons, so for most of these amino acids, more than 1 codon is possible that specifies these amino acids. For example, serine has 4 possible codons here, plus an additional 2 here, so there are 6 possible codons for serine. And others, like valine, have only 4. Some have only 2, like glutamine. And then there's a couple, like tryptophan, that has only 1 codon. There are, in addition to codons that specify amino acids, there are 3 codons, the stop codons, have no amino acid. They do not specify any amino acid. And they're also called nonsense codons by some people. And I'm sure Georgia Tech students will have no trouble remembering that UGA is one of these nonsense codons. The cell actually uses these nonsense codons to specify or signal termination of translation. This is where translation terminates. The opposite of stop codons, then, is a start codon. And there is one codon, the only codon that specifies the amino acid methionine, AUG, is the start codon, here labeled in light green. And this is where ribosomes actually begin to translate. So for the purposes of this course, AUG is the universal start codon. So here it shows how translation starts. We have our messenger RNA template. Okay, it just continues on here. And then the small subunit binds along with an initiator methionine tRNA. So this is a tRNA molecule charged with methionine, and it has the anticodon shown here, CAU, which is complementary to the start codon, AUG. Once that has formed, the large subunit binds, and now we have a complete initiation complex ready to go. What's going to happen next is that the next tRNA, whose anticodon is complementary to this sequence, will come in. deliver its amino acid, so amino acid number 2 will then be attached to methionine. And then the ribosome will move over 3 codons. And this will keep happening. The ribosome will keep moving 3 nucleotides at a time as new tRNAs with their amino acids are brought in. The amino acids number 3 then will get attached to the amino acid number 2. We get continuous peptide bond formation, release of these tRNAs after they have delivered their amino acid. The exit site, tRNAs that end up in the E site or the exit site end up leaving the ribosome. So this process continues, this elongation process continues until the ribosome reaches what's called an in-frame stop codon. So remember, it's moving 3 codons at a time. And then when the next codon turns out to be one of these stop or nonsense codons like UGA, the ribosome stalls, and a protein called a release factor comes in. It fits into this tRNA binding site, and when that happens, the ribosome disassembles, the polypeptide chain is released, and translation stops. So here is the summary, the things that I want you to remember. That translation begins at the 5' end of the messenger RNA, and it always begins with methionine tRNA, so it always starts at the first AUG, and the first amino acid then will be methionine. It reads the messenger RNA from the 5' to 3' direction, 3 bases at a time, and the polypeptide grows from the N terminus to the C terminus. Now, we haven't talked about this explicitly, but remember, amino acids have an amino group and a carboxy terminus and a carboxyl group. Since new amino acids are added to the carboxy terminus of a polypeptide chain, then again, polypeptide chains have a directionality where one end has a free amino group and the other end of the chain has a free carboxy group, and new amino acids are added on to the carboxy group. So polypeptide chains then grow from N terminus to C terminus. And finally, we get termination at stop or nonsense codons. In bacteria and archaea, this is a fairly simple, or, well, I wouldn't say simple, a conceptually straightforward process. There is only the one cellular compartment. There is no nucleus or other membrane-enclosed organelles. So what we see is that transcription and translation are coupled. They happen at the same time and actually are dependent on each other. What we see is RNA polymerase. It has some of the DNA unwound. Here's our template strand. RNA polymerase is proceeding down the template strand, and it's making the messenger RNA from 5' to 3'. And even while RNA polymerase is transcribing, ribosomes are hopping on to the 5' end and making proteins. So this is all happening simultaneously. In eukaryotes, of course, that's impossible. In eukaryotes, transcription and translation are separated in both space and time. In space, because transcription occurs in the nucleus and translation occurs out in the cytoplasm. And time, because it takes time between transcription of the RNA and its processing and exit from the nucleus before ribosomes can get at the messenger RNAs to translate it. What this diagram shows then is the spatial separation. What it doesn't show are additional nuclear processes called pre-mRNA processing. Now I want to make a couple of points here. First, not all transcripts are messenger RNAs. Remember, there are ribosomal RNAs. Their genes are encoded in the DNA, and RNA polymerase makes lots and lots of ribosomal RNAs. So then these ribosomal RNAs, they undergo some processing, but then they go out into the cytoplasm and are assembled to make ribosomes. In addition then to ribosomal RNAs, the next major class of RNAs that cell makes are pre-mRNAs. These are RNAs that encode proteins. In a bacterial cell, there is no processing required. As soon as a messenger RNA is transcribed, it's translated at the same time. In a eukaryotic cell, pre-mRNAs cannot leave the nucleus until they have undergone processing. And this processing occurs at the 5' end, the middle of the RNA, and the 3' end. So the beginning, the middle, and the end of a pre-mRNA is processed in the nucleus before it can leave the nucleus to become into the cytoplasm where it can be translated. So let's start with the first of these processing steps at the 5' end. So remember, this occurs still in the nucleus. And at the 5' end, there's a chemical modification. This unusual modified nucleotide, 7-methylguanosine, is added on with this strange 5' phosphate linkage to the 5' end of the messenger RNA. And you don't really need to remember the structure of this modification. You just have to remember that it occurs, that it's called a cap, and that it is required for eukaryotic ribosomes to initiate translation. So prokaryotic ribosomes don't need any such cap, but eukaryotic ribosomes require this cap to translate an RNA molecule. In the middle, the sequences, if the RNA contains, or if the pre-mRNA contains introns, these introns are removed. What are introns? Well, depending on the species, some or even most genes, protein coding genes, contain not only the DNA sequences that encode amino acids, and these would be called exons. So exons are the sequences that encode amino acids in a protein, but introns are non-coding sequences. I don't want to talk about why they're there at this point, but you have to know that there are these non-coding sequences that interrupt coding sequences, and sometimes they are called intervening sequences for that reason. In any case, these introns have to be removed for the ribosomes to be able to correctly translate these RNA molecules. And this removal process is called RNA splicing, and it occurs in the nucleus and is accomplished by a complex of proteins and small RNAs called a spliceosome. And these spliceosomes are able to recognize the junctions between exons and introns and correctly splice together the exon-exon junctions, and precisely remove all of the non-coding intron sequences. So this shows the intron sequence being spliced out in this lariat form, where it will be degraded into nucleotides for recycling. And what's then left is the RNA where the exon boundaries have been precisely joined to form a continuous protein coding sequence. So we've taken care of the 5' end, which has the cap. In the middle, splicing removes introns. And then the final step is at the 3' end. After RNA polymerase has done its job and the RNA sequence has been made, or the transcription process is complete, another enzyme called poly-A polymerase comes along and just adds about 250 A's at the 3' end. So remember, these A's are not encoded in the DNA. They are simply added after transcription is complete by a separate enzyme that comes on and just adds these A's. What are these? What is this poly-A tail for? It's required for stability in the cytoplasm. What happens is that once these RNAs make it out into the cytoplasm, their poly-A tails gradually shrink, and once the poly-A tail is gone, the rest of the messenger RNA is rapidly degraded. It also appears to have a role, the poly-A tail also appears to have a role in efficient initiation of translation. And finally, the poly-A tail appears to have a role in signaling that the messenger RNA is complete and ready for export from the nucleus to the cytoplasm. So indeed, all three of these steps, the capping, the splicing, and the poly-A tail addition, all occur in the nucleus, and all of these have to occur before the RNA can exit the nucleus and into the cytoplasm. I would like you to view these videos as a way to sort of summarize all of these processes. These are dynamic processes, and it's hard to appreciate what goes on without these animations.
This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene.
These factors are bound to the eukaryotic promoter, and they recruit RNA polymerase.
The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1.
And the way RNA polymerase goes.
The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus into the outer part of the cell.
Remember, it has to undergo processing first.
In a dazzling display of choreography, all the components of another molecular machine locked together around the RNA.
There comes a small subunit,
called a ribosome.
large subunit.
It translates the genetic information in the RNA into a string of amino acids that will become a protein.
You can see the aminoacyl tRNAs entering the ribosome.
The green triangles bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. ribosomes for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.